Hello everyone, my name is Abdu, and today I'm excited to show you how natural mutations in the movement of protein of soybean vein necrosis virus was able to allow the protein to express and delay the cell death in the plants. So first of all, SVNV or soybean vein necrosis virus is a single-stranded RNA virus that was first discovered in Tennessee in 2009. This virus belonged to a bigger group called orthotaspoviruses, and all of these viruses, their genome is composed of three segments, as we can see here on this figure. So we have S and M and L segments. Today, we are going to be focused on one protein on the M segment called the movement protein or the NSM protein. So it's important to study these viruses because, as we can see on the phylogenetic tree, SVNV have an interesting clustering on the phylogeny tree. So it clusters within one clade called was B necrotic mosaic virus. And that's interesting because it indicates some variations in the virus sequence or maybe the infection cycle that we didn't know about today. As I mentioned, this virus was recently discovered, so we still don't know a lot about it. And we've done a lot of work on it over the past 2 years that we have revealed a lot of information about this virus. And today we're going to just talk about a tiny findings of ours. So as I mentioned, we're going to be focusing on the NSM protein, which stands for movement protein. And as it's called, it allows the virus to move from cell to another cell during the infection. So NSM protein, a little bit of background about this protein, it belongs to a bigger family called the 30K superfamily. So why they named it this way? Basically, it's a group of divergent viruses from different families, and their movement protein makes about 30 kilodalton, so they named them 30K. And basically, they have conserved domains and motifs that they share among each other. That's why they put them in the same group, and they function as movement protein. However, the orthotaspovirus's movement protein have another, or beside that they have another thing to do, which is something called tubule formation. So tubule formation is a domain that they contain, and it allows them to form tubules. How this happens is that the protein self-interact with itself and it forms tubule to allow the virus to move from cell to another cell. And this is something fascinating about these viruses. So what is the gap of this work? So basically, we don't know what is the cause of necrosis or how the symptoms of necrosis are being developed or what protein from the virus is causing necrosis. So this is the gap of the work, and hopefully we can reveal it today. But we have an previous information that helps us to focus on NSM or to choose NSM for this study. This information that we have from the past is that TSWV, which is one of the main known viruses in orthotaspoviruses, its NSM, it has been linked to the symptoms development of this virus. Therefore, we said, okay, maybe NSM of this virus also causes necrosis, or it might be causing necrosis based on the previous information. And we wanted to test that hypothesis. So basically, my hypothesis is that natural mutation in NSM will affect both the expression and it could be contributing to the development of necrosis symptoms in plants. And I have two objectives to investigate my hypothesis. The first one is to analyze the functions of NSM of SVNV and also the mutation that we found during sequencing. The second one is to localize NSM mutants in the plant cells to determine if they have any potential in causing necrosis or delaying the necrosis, as we will see. The method that I use is that basically I use the PCR to amplify all the mutants, and then I clone them in the pENTer. Then I send them for sequencing to make sure that the sequence is good. And then once I get the sequence, I did two things. I did the domain analysis and I did the fusion into the P site, which is a good plant expression vector. And in P site, the proteins are being fused with GFP from both terminal C and N terminal. Then I did agrobacterium transformation, and agrobacterium is the shuttle that's going to deliver my plasmid to the cell. And then finally, I did infiltration in Nucleotiana benthamiana, which is a host for SVNV. So it's a good expression plant for this virus. And also after that, I've done the confocal imaging, or basically the microscopy imaging using the confocal microscope that we have. So for the results, as you can see here in this domain analysis, I compared NSM of SVNV to other orthotaspoviruses, including BNMV or B necrotic mosaic virus, TSWV, INSV, IYSV, CACV. And all of them, they contain the domains that are, not all of them basically, some of them would contain all the domains that are found in the 30K superfamily, such as TSWV, it has the 4 domains. However, SVNV, it has only 2 domains, the T motif, the D motif, and the G residue. Beside that, also all of them contain the tubule formation domain. As I mentioned, they have this advantage over the other 30K superfamily, or a different advantage, or basically a different function. However, there was one domain that was unique to SVNV, and it's called DUF, stands for domain of unknown function. And this is very interesting, and we will try to talk about it today. So this is how the domain looked like of SVNV compared to other orthotypical viruses. Here, I also wanted to make more analysis about the two samples that I localized in my study today. So I have the NSM of the wild type. This is the Tennessee sequence that was first isolated in 2009. So it's the original sequence that we first have about this virus. So NSM in 2009 looked like that. It did not have any mutation. It has both the domains, the DUF and the tubule formation domain. The sequence from Alabama, 2023, which is 15 years after the original sequence, and it contains the same two domains. However, it has a mutation in the middle, almost in the middle of the protein. This mutation where the valine changed to isoleucine in this position, which 143. So valine and isoleucine, the change is still within the same group of the amino acid. It did not change the group of the amino acid. And this mutation appears with 42% in the population. So about 40% of the population of the 33 samples show this change. And I wanted to see if this mutation will impact the folding or the conformation of the protein or not. And not to lie to you, before I do that, I thought it's not going to cause any changes because it's just one mutation. It's a point mutation and it doesn't even change the group of the protein, of the amino acid. However, I was shocked when I used the AlphaFold 2 to predict the structure of the protein, and I found that, however, just to be also honest, this side here, it's appeared in this color because it's very low confidence. However, it shows us changes between both of them. So we look at the wild type, we see that it's unfolded. The protein terminals, both of them are open. They're not folded to each other. However, the one with one mutation from Alabama, it is have a proper folding to itself or to its core. And we will talk about this in like a few minutes. So what happened when we put both of them in the plant? When I expressed them in the plant, so just to help you understand this figure, the first column is GFP fused to NSM. The second column is the plant. It was a transgenic RPER plant. And also after that, I used cytox, and I will explain why I use cytox. And then we have overlay and then a large section to show you what's happening on the level on 1 or 2 cells. So basically, the NSM of Tennessee did not express at all at 2 days post-infiltration or at 4 days post-infiltration. There was no expression. However, the NSM of Alabama was able to express and show us a good level of expression. My control here was N, which is another protein from the virus that we know it can express and it doesn't cause cell death. So why I use cytox? Because when I infiltrated with NSM of Tennessee, after at least 10 replicates, I've never seen NSM express. And I was desperate. I didn't know what's going on. However, I started observing symptoms of necrosis, or basically cell death. This appeared to me with my naked eye as patches of dead cells on the infiltrated leaves. But we want to confirm if this is cell death or not. So I use cytox. Cytox is a stain that basically it tests the cell viability. If you can see this stain inside the cell, it means those cells are dead because the stain cannot penetrate their cell wall if they were in a good shape. Once this stain penetrates the cell wall, it binds to the DNA and RNA in the cell. And if you see it inside, as I mentioned, it indicates cell death. So at 2 days post-infiltration, we see that none of our candidates cause cell deaths. You can see that the blue stain is all outside the cell walls. However, at 4 days post-infiltration, we see that the stain in the case of the Tennessee NSM is now inside the cells, indicating cell death in Tennessee NSM. However, the NSM of Alabama was with the mutation did not cause cell death at 4 days post-infiltration. So what are these results mean? So when we go back to the folding and unfolding, basically, I looked at this paper, which is a beautiful paper about the folding and unfolding of the protein. It has more than 10,000 citations. And basically the conclusion that I got from it is that when you have a protein that is unfolded, doesn't have a proper folding, first of all, it might not be able to express normally. It will trigger bad things in the cell, such as cell death, and it might not be even functioning. And that's what we maybe can see here in NSM of Tennessee. It doesn't have a proper folding, it's open, and then it did not express and it caused cell death. However, the NSM of Alabama with one mutation, it has more proper folding to itself. This is to protect the protein and also to allow self-interaction. So it allowed it to express and delay the cell death. So today we revealed that, yes, NSM of SVNV is the one causes necrosis because as we've seen here, we have cell death at 4 days post-infiltration. Now, if you ask me, Does even the mutant still cause cell death? I believe yes, it is still causing cell death. So what's the difference? So the difference now is that one of them is causing cell death really early in infection, but the other one, which is the mutant, is delaying this cell death. How is this important for the virus? Basically, if you kill the cells really early in the infection, you're not doing the right thing. You're in trouble because you kill the cells before you leave from the cells, or maybe before a good numbers of you leave the cell. So basically, you're not very successful. However, when the virus delay the cell death, it can then spread to other cells before the cells that were infected in the first place dies. So this is what the evolution of the virus looked like, and this is how it's evolving to enhance its function. So as a conclusion, a single mutation in the 3A domain, which is the tuple formation domain, was able to allow the virus to express and delay the cell death. There's a lot to come about this domain that we don't know the function for it yet, which is the DUF domain. Basically, I wanted just to mention that it comes from bacteria. It was originally found in bacteria, such as Clostridium species, and it's very interesting to me how this domain got into SVNV NSm. There's a lot to reveal about this domain. We cannot say a lot about it today because we're still investigating what is the role of this domain and if it's the only one actually responsible for this cell death or not, and maybe the proper folding here, as we can see both terminals here folded to each other. Maybe this mutation, however, did not happen in that domain. Maybe it's still impacting the function of that domain. So if this domain is really strong, the unknown domain, if it's really strong, it might trigger cell death. However, with this mutation allowed proper folding, maybe it's reducing the cell death of this domain. We don't know yet, but this domain is very interesting to discover and to talk about it in the future. So for future work, I have to localize the rest of the mutants because I have other mutants as well. I just localized one of them today, and also to test the expression of these proteins using the QRT-PCR, and then finally to analyze the expression of the plant genes after the infiltration as well to test what is the plant reaction to those proteins. And as acknowledgement, I would like to thank Auburn University and our PI, Dr. Martin, and the Soybean board for funding and supporting this work. And thank you so much. And I would like to have any questions from you. You can email me through my email. Thank you so much and goodbye.